Study of fungal.
Study of fungal. Okay. Before the explanation, here mycology in the plant systematic position. Non-flowering plants, you know what is the non-flowering plants. If you can see the my PPT, PDF notes. Yes.
Yes, ma'am.
Yeah, thank you. See, before that one here today, I'll explain to the what is mycology, what is the study of fungi, what are the names, what is the structure of fungus. These are things we will discuss today. Okay. And in this one, the first before will explanation, what is the fungi? First one is the what is the fungus is there. As a present biologist, the term use the term fungus. Fungus is the Greek word. That one is a Greek word. Meaning of fungus is the mushrooms from sponginous, the spongy. Okay, the total fungus is the fungi meaning mushrooms, sponge, the name. It includes eukaryotic, spores bearing, aclophilic organism. Okay, this one is the main is the, you can observe the eukaryotic cell is present in this one. Here, organisms of fungi generally reproduced sexual reproduction and asexual reproduction. Usually filamentous plant body. What is the plant body is there? Filamentous, branched, stomatic structure are typically surrounded by a cell wall. Containing here another very important, the plant body, you can observe the in the algae, plant body is a thallus, but here the plant body is called the that is a mycelium. The plant body name is called mycelium. This is a Greek word. The fungus meaning mushrooms sponge. The total, the meaning is the mushrooms sponge. And reproduction, you can observe here, sexual reproduction and asexual reproduction. Whose usually here filamentous bodies are there, branched, stomatic structures are typically surrounded by cell wall. Another very most important cell wall here, the cell wall main important, the main important is the made up of the chitin. The cell wall is made important to the chitin. That is the most important. Many times to competitive exams to ask the question, how to the fungal cell wall to made up of chitin or celluloses, or both of these substance together with many other complex organic molecules also. The basic cell wall is the chitin and or cellulose and other substance also, other organic molecular also found. In formal and concise way, fungi may thus be defined as, these are a character based on the fungi, defined as eukaryotic characteristic, mycelium, mycelial heterotropous. See, these are the heterotropous. What is the meaning of heterotropous? They depend the food material other organisms. In algae, eutrophous. But fungi is the heterotropous. Why heterotropous? They do not prepare the own food material. They depend the food material other organism. That's why the fungal body name is called mycelium and heterotropous. However, organism regard as fungi are very, very variable in form, behavior, and life cycle. For instance, usually organisms like myxomycetes and similar forms are not filamentous but are traditionally included among fungi. In a few others, the filaments have no cell wall. Where the filaments are there? In that one, no cell wall. And it is thus difficult to give a concise definition of fungi. Main the important scientist name, keep in your mind, Ainsworth. The main explanation of total fungal explanation or father of, or just like the scientist name is called Ainsworth. The year 1973, therefore, preferred to enumerate the some characters. And Ainsworth to enumerate the this is fungi. They explain to the some character. What character? Here, first one is the nutrition. These all the heterotropism. Photosynthesis is lacking, and absorptive uh, ingestion rare. That is a nutrition. Thallus, what type of thallus is there? Thallus on or in the substratum and plasmodial, amoeboid or pseudomycelium in the substratum, and unicellular or filamentous. The last septae or non-septae, typical non-motile. All others, some but few are motile. Where the non-motile is there, with protoplasmic flow through the mycelium. But motile state is called the zoospores. This is the thallus. What is the cell wall? Typically chitin, cellulose, oomycetes. The basic thing of the cell wall is made up of chitin. Some oomycetes order is there, no? In that order, cell wall to made up of cellulose. Nuclear status. This is the eukaryotic type of nucleus. What type of nucleus is there in this one? Eukaryotic. See, this is the eukaryotic type of nucleus. Multinucleate. The mycelium being homo or heterokaryotic. Haploid. Dikaryotic or diplotic. Limited duration. Life cycle very simple to complex life cycle is there. Sexual reproduction, sexuality, asexual and both sexual. Sometimes homosexual or heterothallisms also present in sexual in the reproduction time. Sporocarp, microspores, macrosporic, showing limited tissue differentiated. And next one, habitat, ubiquitous as saprops, symbiotes, parasites, hyperparasite, distribution cosmopolitan. This is the total. What are the fungi? I will explain. Just what are the fungi are there? What is the importance of fungi? I'll explain. We will discuss about the economic importance of fungi last class. Okay. Before today, first one general. What is the general account of fungi? Main important, what is the main general account of the fungi is there? How many fungal related to the talus structure? These are things are we will discuss. Okay. Another important character here, fungal main, this is the thallophyta, no? That's where the thallophyta you can't observe to the directly in the eye contact. You can observe in the main source of the under electron microscope. In the fungi, the first one is the cell structure. Fungal cells may be minute or single. Only single nucleate is present. That's why uninucleate cells that may constitute entire organism. The single cell, olphidium or yeast cell. Yeast is the best example. Single cell. Keep in your mind, single cell fungal member is called the yeast. Fall in this category. Alternatively, fungal cells may be elongated and strand-like. Several may jointed to the thread-like cells are present. That thread-like form is called the hyphae. See, the cells may be very single, multicellular is there. Uninucleate to multicellular is present in the fungi. Unicellulate or unicell structure, best example, yeast. Another one is the elongated strand-like cells. Several may be jointed. Thread-like cells are there. In that thread-like cells name is called hyphae. A large number of, in that one, next point, a large number of hyphae that the combine with the group of hyphae collectively to form to the one portion that is called the structure. That structure name is called the mycelium. And here, main important, the fungal plant body name is called mycelium. The cell wall fungi generally, I'll explain now before, generally made up of cellulose and chitin or chitin. Cellulose or chitin. Major group of fungal to made up of the cell wall chitin only. Occasionally, chitin wall, occasionally, sometimes you can observe to the both the cell wall to made up of the chitin and cellulose may occur simultaneously. This is the important. The basic point, the study of fungi is called mycology. Okay. Next one, the cell wall. Coming to the cell wall. This is the heterotrophism, not eutrophism. Heterotrophism meaning they do not prepare the food material own. They prepare the food. They depend in the food material, that is the heterotrophism. And the next important characters is fungi cell wall. What is the cell wall? Main cell wall made up of the cellulose is common in algal, but this is the second step of the plant body, no? That's why this one are called the cell wall is made up of chitin. This is the basic thing. But occasionally, second one, cellulose also. Sometimes you can observe both cell wall to made up of, that is a chitin and cellulose. Did you understand my topic? Anyone?
Yes, madam.
Yeah, thank you. Simultaneously. See, in case the plant body, my fungi also just like one plant body. The one cell in hyphae is there, may be separate from other cross walls. That is called a septum. One hyphae separate from another cross walls are there. That name is called a septum. Here, septa in different group of fungi have important difference in structures also. But for example, what is a septa? What is that one? In case, example for the septa, in the lower fungi, here the starting fungi is the, after algae, the second one is the fungi, no? The lower fungi, here, main important of the lowest or lower fungi, pseudoseptae. What is that? That is called all the pseudoseptae, which are perforated by the so many pores you can observe, resemble like a sieves. And this is the first example. Another one, ascomycota. The pseudomycotina. Here, in the fungi, first one is the classes are there. One is the masticomycotina, ascomycotina, zygomycotina, basidiomycotina, and deuteromycotina. These are the classification. When the classification time, I'll explain that one. Okay, in the ascomycotina, fungi, some fungi, imperfectae. The septa is the imperfectae. The septum perforated in the only one pore is there. That is a single pore. That one is the imperfect type fungi. Or in that hyphae or in that septum is the like that. The septum is more complex than that found in the ascomycotina. This type of septum is called here dilophore's septum. Very important. In the study of the type studies, in that time I'll explain to the clearly what is the dilophore septums. Okay, coming to the hyphae characters here, grow at the tip and can usually produce lateral branches. There are old parts. The mycelium may remain microscopic. You can't see in the directly. You can observe to the in case in the pathology time, in some plants causing the disease fungal. In that time, you can section it under electron microscope. You can observe to the spores very clearly. How to observe? Only under electron microscope. Yes or no? That's
నాకు ప్రతి సమస్యకు ఒక సవాల్ ఎదురైంది సార్.
ఎగ్జామ్ వచ్చిన
ప్రతి సమస్య పెద్ద సవాల్ ఎదురైంది. ఎందుకంటే ఇలా కాకుండా నా భార్య ఇష్టంతో వెళ్ళిపోతుంది. ఇష్టంతో వెళ్ళిపోతుంది మా ఇంట్లో దాంతోపాటు
the interruption. My network issue is there. That's why. Okay. One second. Again, I will share my PPT. Yeah. Did you observe now? Yes. See,
Yes, madam.
yeah, okay, ma'am. In this one, hyphae characters, main important, the fungal body is the microscopic structure. Microscopic structure. And next one coming to this part, hyphae is the characteristic features of this one. And next part of the hyphal, fungus tissues is the most important. The mycelium remain into the macroscopic structure. Easily organizing structures are present. Okay, in the fungal tissues made up of prosenchyma and pseudoparenchyma. You can observe the fungal plant body. Mycelium is there, but this mycelium is very smooth mycelium. And observe the total tissues made up of from the parenchymatous and pseudoparenchymatous tissues are present in the body. Here, next one, the nuclei have each and every cell you can observe to the all growing hyphae with cytoplasm is there, and frequently vacuoles are also present. Maybe aseptae, all are the aseptae conditions. And a single nucleus is there, that is a cenocytic. One single multinucleate cell, single multinucleate. Single multinucleated meaning is called the cenocytic bodies. That name is called the cenocytic bodies, are divided into segments by the septa. These fungal nuclei very, very small. The nuclei have well-developed nuclear membrane. The nuclear envelope consists of double nuclear membrane also to present. During the mitosis, the nuclear envelope does not nuclear envelope does not disappear ordinary, and instead of consist the dumbbelled like fascinations are present in that one. And in the nucleus you can observe the meiosis is there, and after meiosis, here the other organisms also does be mitosis, only this one. Another very important in fungi, ribosomes you can observe. Here 80S type of ribosomes is present, and mitochondria occurs, you can observe to the in the cytoplasm. There is also the endoplasmic reticulum. Storage productions occurs in fungal cells, and you can observe the each and every cell contain type of ribosomes, 80 type of ribosomes. In the cytoplasma, you can observe to the mitochondria and endoplasmic reticulum also. They may be in the form of here the food material to fix in the production in the cells. That is a storage material. This storage material is the food material stored with the glycogens or lipids. What type of food material to store? Glycogens and lipids. And another cell organelles, that is called the Golgi complex. Golgi operators in fungi, similar to those in the higher plants, animals, have been found in the some fungi also. These are features which is the widespread occurring in the presence of the lysosomes. These are made up of membrane bounded tubules or vesicles are present. In some fungi, this lysosome is not known in the fungi. These are the total cell structure. What is the cell? How to the structure is present. Coming to the reproduction. In the fungi, reproduction, only two types of reproduction you can observe. Both the asexual reproduction and sexual reproduction. Two types of reproductions you can observe to the total fungus. Main important here, spores, that is the most important characteristic for the spores also. Spore is the asexual and sexual both reproductions to present total in the fungal plant bodies. And next one here, what is the asexual reproduction takes place? Here, different types of asexual reproductions to present. Main asexual reproduction is called the formation of the spores. Spore structures is the most important for the reproductive structures. Here, main important, it's a mycota. First one, fragmentation of the soma. Second, asexual reproduction, fusion of stomatic cell. Third type, budding of the stomatic cell. Fourth type, production of spores. This spores is called conidia, sporangiospores. These conidiospores are sporangiospores, motile or non-motile part. These four you can observe to the asexual reproduction parts and different types of asexual reproduction. Coming to the sexual reproduction, widespread occurring in the fungal, fertilization when brought to about in various of varieties of way. The first one, planogametic population. The first sexual reproduction, planogametic population. Second one, gametangial contact. The second one is the gametangial contacts. Third one, gametangial copulation. Fourth one, spermatization. Fifth one, stomatogamy. Stomatogamy is the fifth one. These are the reproduction two methods, asexual, sexual. Asexual methods, formation by four types. Sexual method, like these five types of are very common. Coming to the life cycle. See, as in other organisms, the fungi, there is a general cycle is present, that is a haploid and diploid structure. Haploid meaning only single one. Diploid in both are present, male and that is a diploid condition. Here, corresponding to the gametophytic and sporophytic in green plants. What is the gametophytic generation? Sporophytic generation in all green plants. The diploid phase begin with karyogamy. What is the meaning of karyogamy? Anyone tell me? Anyone? What is the meaning of karyogamy?
The fusion of nuclei?
The fusion of?
Fuse. Nuclei.
Fusion of nuclei kaadu. Here, in the nucleus, nucleoplasm is there, no? Yes. In that nucleoplasm, divisions are there. In that one, karyokinesis. Okay. In the cytoplasm, divisions are there. In that one is cytokinesis. Okay. Did you understood? Karyogamy, karyokinesis and cytokinesis. In the nucleus, division, nucleoplasm divisions, karyokinesis, cytoplasmic division, cytokinesis. Here begins with the karyogamy and ends with the meiosis division. The majority of the fungi, there is no distinct alternation of generation. Very, very important. In algae, you can observe the many alternation of generations are present. But there is in fungi, major 99% or 90% group of fungi, there is no distinct alternation of generation. The scientist Raper in 1954 recognized 7 basic type of life cycles in fungi. Another scientist, Bernetti, in 1976 recognized 5 basic life cycles of fungi. Namely, to both scientists is the most important. One Raper, another one Burnett. In Raper is the 1954, Burnett scientist to explain into the 1976. Here, the first cycle, asexual reproduction in which sexual reproduction is lacking, namely deuteromycetes. Okay, did you understand? Asexually, in which sexual reproduction is lacking, where in Deuteromycotina. Well, when I'll explain to the classification time, in that time also I'll explain. In Deuteromycotina, you can observe only asexual reproduction. Which one is lacking? Which one is lacking in Deuteromycotina? You interact with
Sexual reproduction,
Sexual
ma'am.
reproduction.
sexual. Very important in competitive exam. Ask this question. I'll give the example later. Deuteromycotina. In that Deuteromycotina, what type of sexual reproduction spores are present to ask? But in the all Deuteromycotina, sexual reproduction is less, lacking or absent. Only you can observe which one? Which one to observe? Only asexual reproduction. See, you can observe to the this, next one. The second one I'll explain to the
Reproduction.
okay. Next one is the Ascomycotina. Here, haploid in which meiosis immediately followed by the nuclear fission. This is very common seen in fungi or some members of Ascomycotina. The diploid phase is the minimum duration. Okay, what is the condition? Next one is the haploid in which meiosis. Haploid condition. You can observe the picture. Okay, this is the ascospores. Okay, these are the ascospores. These ascospore present the ascospores. In this ascospore, you can observe the germinate the ascospore. Each and every ascospore after germinate, you can make it matting structure is there. See, this is the matting type A. This matting type A. Both are the combined with the, this name is called plasmogamy. Combination, combined with both are matting structure. These both combination is called the plasmogamy. The only plasmogamy, the cell wall to rupture and that part is to enter into the both cells. That is called the plasmogamy. After plasmogamy, you can observe to the 2 branches like myceliums are there. This name is called the dikaryotic mycelium. N plus N is equal to 2N. Yes or no? Dikaryotic dikaryotic mycelium is present. But this is a haploid hyphae. Why? Only N character, N phase chromosomes are present. That's where this is the karyotic haploid hyphae. In the haploid hyphae, after development, you can observe where my arrow mark is there, that one you can observe. This is the ascus or ascocarp. This is the fruiting body. In that fruiting body, you can upper part, the light dark color is there, no? That is called to produce the ascocarp, the ascospore. See, like this. This is a dikaryotic acai. Ascocarp is the fruiting body. Ascospores, the finally to produce the SI, ASCI. This is a haploid condition. After this one, after fertilization of this, karyogamy fuse with nucleus. In that one, this fused nucleus after this one to meiosis, undergoes the meiosis. What happened meiosis? the reduce the nucleus. Yes or no? Reduce the nucleus, and this one to undergo to the mitotic division. That's why this is the haploid in which immediately follows to the follows of nuclear fusions. There is very commonly seen in fungi, that is the Ascomycotina. This is the minimum or duration of the diploid phase. This is the second thing. The third one you can observe to the third picture here, haploid dikaryotic. Which one is there? Haploid dikaryotic, which is similar to the second type. Just like the similar part. This one is a similar to the second type. Expect that paired potentially conjugate nuclei persist close physical occasion in the same hyphal segment. Hence, this is called the dikaryon. This part is called the dikaryon. You can observe the picture. I'll enlarge, no? See, this is the dikaryon. This one, you can observe this part. Mother cell of the gametes or meiospore. This name is called the gametes or meiospore. In that one, you can observe to the, this is the interphase. You can observe this phase is called the interphase. After divisions, interphase, here 4n is there. Interphase in the G2 phase, you know that one. After interphase, the divisions to start, that is the prophase 1, metaphase 1, anaphase 1, telophase 1. Again, prophase. This is the meiosis division, no? Two stages are there. Meiosis 1, meiosis 2. Meiosis 1, this is the first interphase division, this one. This interphase, you can observe the here total interphase, how many phase? Prophase 1 is the many phases are there. Diplokinesis, karyokinesis, diakinesis, like that. Plectotin, leptotin, like this. After this one, prophase 1, prophase, metaphase 1, anaphase 1, telophase 1. After telophase, that second meiotic division to start, that is the prophase 2, metaphase 2, anaphase 2, telophase 2. After telophase, you can observe to the, see, you carry, this is the cytokinesis divisions to start here. This one to produce one zygote, this one to produce another zygote, this one to produce zygote, this one to produce zygote. These all are called gametes, plants and fungi. That is called the one second. That is called the gametophytic stage. Here, haploid dikaryotic, which is similar to the second type of the life cycle. Expect that paired potential conjugate nuclei persist in close physical occasion in the same hyphal ring. That is called dikaryon. For example, many members of Ascomycotina binucleate ascogenous hyphae develop from the period to ascus development. Such dikaryon can't exist independently. This dikaryon never independently can't exist. That's where the another, and there may be condition where the meiospores. Okay, these meiospores in the dikaryosis accept the movement of fertilization. For example, in yeast, this occurs, you can observe in the yeast only, uninucleate, that is the endomycelates, more commonly seen in smut. So what is a smut here? Here fungal causing the many types of diseases. Smut, rust diseases. You can observe the smut eustilaginales order. This one also I will explain to later. Where the smut diseases are there, smuts are present, that one all eustilaginales order, you can observe the smut. In the Basidiomycotina, the mycelium derived from the germination of a Meiospores may persist in the haploid condition as monokaryon. But once a dikaryon is formed, it shows unistricted independent growth is there. That's why this life cycle name is long-lived phase of life cycle. The second one is called the long-lived phase of the life cycle. Coming to the third one. Next, fourth one. See, this is a nuclear fusion. Outer nuclear fusion, you can observe that one. This is the nucleus. And to fuse after that one gametes, 2n stage. After 2n stage, meiosis division is there. Two off of the cut is there. That is the two offs. And the next one, vacuoles. This is the vacuole. And this is the stigmas. What is this one? It's stigmas. In this stigmas, these are nuclear fusions to coming to the stigmas. This is called the basidiospore. This one vacuole to increase in size, that is called basidium. When the reproduction time, I'll explain. Okay. Coming to the another one, fourth life cycle, haploid, diploid, in which the haploid and diploid phases is both are alternative phase. This you can observe only blastocladiales order. In the very rare this one. Haploid and diploid both are present in only one order. That too you can observe to the very regularly, alternately you can observe. The order name is called blastocladiales is the order. See, this is the alternation of generation you can observe. These are the spores. Spores always end stage. Okay. These spores after mitotic division to form many. In case 4, 8, 16, 32 like this. This is the many spores to form. This is the gametophytic stage. Each one you can observe, each one is called the gamete. These gamete fuse to form to the zygote. This is a 2n stage. Yes, no? Yes or no? Always zygote is a 2n stage. These zygote after mitosis division to form the sporophytic stage. This sporophyte after division of the meiosis to cut the off of the cells, no? Meiosis to form again to the spore. This is called haploid and diploid alternate regularly. This is the very you can observe. Did you understood? This is the haploid condition. This one diploid condition. Both are to form to the regularly and restricted to the order, only single order. That too blastocladiales is the order. Coming to the fifth one. See here, in fifth one, diploid in which the haploid phase is restricted to the gamete. This is the first one thing is a diploid. But in which the haploid phase is restricted to the gametes. The vegetative phase is the diploid phase. The reduction takes place by the time of gametogenesis. Example for Phytium, Phytophthora, Sporolengia species, Mycetes fungi. This condition you can observe the type of life cycle. Phytium 1, Phytophthora indica, causing the white rust disease in crucifery family, and Sporolengia species, and Mycetes fungi. You can observe this type of life cycle. See, here the light color, bluish color, light color is there. These are the haploid condition. Light violet color is the heterokaryotic. Unfused nuclei form different parents. Light pink color, diploid condition. See, spore produced structure. Mycelium is the first one. This mycelium to produce the spores. These spores germinate. And after germination, spores to form to the plasmogamy. This is the fuse of cytoplasm and form to the heterokaryotic stage. This karyogamy, under the after karyogamy, fuse of the nuclei to form to the zygote. When the zygote to start, in that time to another division is the meiosis division. After meiosis division to formation of spores. This is the basic life cycle of the fungal plant body. Coming to the, this is the total life cycle and reproduction. Okay, did you understand today reproduction and life cycle? Yes?
Yes.
Yeah. I'll take till 1 o'clock. After that one, I can leave. Okay, ma? Yes?
Okay, madam.
Yeah. Coming to the next one is the classification. Very, very, very important. Classification, based on the classification, the fungal is the, some characters are the most important to classify the fungi. Minimum here, the Whitaker, the 1969, the scientist name, most important in sometimes to ask the scientist names also in the competitive exam time, not for the descriptive time. When you can write down the competitive exam, in that time you can explain to the, you can by hand it to the these scientist names also. For the main important, for the past 60 years or so, most classification of the fungi have differentiated the characters. Plasmodial. Plasmodial meaning myxomycota, wall-less form from the mycelium. That is the Eumycota, true walled fungi. Okay, did you understood? For the past 60 years, or so most classification of the fungi here differentiated the plasmodium, plasmodial. Plasmodial meaning this is a myxomycota. Under myxomycota, you can observe the plasmodial. This myxomycota wall less forms. And the second one, mycelial. This is a eumycota. True cell wall of fungi is present. There are three natural classes and one from class of true fungi have been universally recognized since the early days of mycology. The classes based on the type of sexual produced spores. This is the most important. The scientist is the Whitaker in 1969, and Whitaker 1969 broken the tradition of a three kingdom system of classification, living organism. Recognized that the classification living organism is called prokaryote, animals, and plants. Here, plants you can observe. Okay? The Whitaker scientist, total living organism divided in three. One is the prokaryotes, you know, bacteria, virus is the prokaryote, mycoplasma. The second thing, animals. All animals include human beings also. And plants. Here, plants, fungi also plant. You can't the animal, you can't that one. Fungi also, including plant, did not reflect their relationship. With Whitaker added two more kingdoms in the classification that is called fungi and protista. What is that one? Fungi and protista. In the fungi, uh, Guinea wagon and Barnus, 1926. Guinea wagon and Barnus in 1926 did not consider the myxomycetes as fungi. These appear to them to have arise separately from the protista. Myxomycetes were included by them, form resembling fungi. The fungi were divided into four classes on the basis of nature of vegetative thallus characters. Okay, different scientists to explain different ways, ma. Okay, the first kingdom fungi Chytrodimycota. That is the Zygomycota, Glameromycota, Ascomycota, Basidiomycota, Mytosporic fungi. Okay, this is the one type of phyla. Another six phyla. This one, Straphinophila, Chromophysta include in three phyla. That is the Oomycetes, Hypochytridiomycota, Labyrintholomycota. And another four phyla kingdom slime molds are present. This is the Myxomycota, Plasmodioformomycota, Dictyosteliomycota, Archisiomycota. This is the based on the six phyla that is called the main important is the molds or spores. See, this is the this one is the main a brief introduction of the fungi. Fungus like organisms. The fungi is the Mycota or Eumycota. Mycota is the main important to the main Mycota, and second one is the Straminiphilia, and third one is the slime molds. These are the divisions. And what is the Mycota? Eumycota is there. In that part, Mycota and Eumycota, here main important of the Mycota, Eumycota, Cryptolodimycota. And next one is the Zygomycota. Next one is the Zygomycota. Next one is the Glimoromycota. Next one is the Ascomycotina. Next one is the Ascomycotina. Next one is Basidiomycotina. Next, Mytosporic fungi. This is the main important of the fungal one. Okay. Next one is called...
Madam, differentiation between mycota and eumycota, madam?
Yeah, I'll explain later. What is the mycota? Later, I will explain. This is the starting.
Okay.
When I'll start, no, in that time, I'll explain what is the mycota, what is the mycota. Okay.
Okay, okay.
Okay. This is a total fungal-like organism, not fungus. No, I'll explain. Fungal-like organisms, the classification like this. Okay. Next.
Okay, okay.
Okay, now. Different phyla and subphyla is fungi. Based on the, you can observe the brief description, this fungi is the types. These are the different types of the fungi. In the year, Mycota fungi, I can explain in the brief description. In this one second, ma. Here, main, this is the Mycota and Fungota. Before in that one, I'll explain the ones here. Here, Mycota is there, no ma? This part, 1 second. You can observe this is the Mycota. And Mycota meaning, Mycota meaning true-walled fungi is the Mycota. What is the Mycota? True wall fungi is present. It is called the Mycota. Mycota meaning this is here. For true wall fungi is totally absent. Sometimes you can observe to the Mycota. Okay, that's where this is called the main difference in between the Mycota and Mycota. Here
Okay, ma'am.
Okay. Next one, coming to the Mycota, main Mycota, were divided in three natural classes. Phycomycetes, Ascomycetes, Basidiomycetes, and different scientists different way to explain to the fungal. Okay. Another coming to this one. This is a mycota. Okay. This one is a mycota. Here, uh, Chytidomycetes, very smallest, oldest, simplest fungi. This one is motile cells are present. Motile, you know, that is the flagella is present. Where you can observe flagella, zoospores and gametes, and single posterior flagella and cenocytic thallus is present. The cell wall contained to the chitin, glucan, nuclear division, intranuclear, and centric. These characteristic is called the mycota. You can observe this one. Cell wall is present, but it is not true cell wall. Mycota is a true walled fungi. Did you understood now? Uh, Mycota characters are these. See, this is an example for the mycota. Spicigellomycetes. This one is a spicigellomycete. You can observe the juice spores with several lipid globules, and sometimes you can observe the cell amoeboid motions are there. Rhizoids also present. The each and spore you can observe to the minimum 5 nanometers in diameter, and predominantly from form to the soil. Some important genera also that is there. Olphidium orders are there in that one. Olphidium, Rosella, and Callocrium. These are the orders. The second class, Neoclamisticales. Neoclamisticales, see, you can observe anaerobic chytrids that inhabit the rumen, and also this is the main important you can observe in that part. That is the most important characteristic features of the in this fungi. Here inhabit rumen, and also this the characteristic also animal characteristic is present. That characteristic feature is called the that is the herbivorous type of the characters are present. Next, coming to the, they are placed in 5 genera, include the neoclamistics. See, this is a picture. Next one, Tritidales. They are waters. You can observe this type of fungi. They are water, soil inhabiting fungi. Many of them farmer parasiting on algae. Very important. These fungi parasitic on the algae also. And this is the water. You can observe to the water molds. Where the water moles is there, in that part you can observe the water moles, and later vascular plants, and next one few parasitisms are there. This all are the you can observe to the this type of fungi. And next one, coming to the exam orders for some better known to this one: Critidium, Critodismus, Endochitin, Synchritidium, Cladocritidin is the best order from in this part. Next one, coming to the another vein, another second one, Zygomycota. Zygomycota is the blasticales, and the coming to the Zygomycotina, and this one is the Ascomycotina. In this one, Zygomycotina, you can observe these molds. You can observe all type of molds that live in the soil, or decaying or plants, animal material observed to the this Zygomycotina. Here order Blastocaldiales. This order chiefly main water and soil, that one inhabiting fungi. Blastocadals genera characteristic thick-walled resistant sporangias are present. Pitted walls also present. Another very important feature, linking membranes also, predominant nuclear cap also present. This one, the different families are there. That is the blastocardians, phycocermidales. These are the class. And the next one is the mostly another one is this part, trichoderma. Mostly trichoderma you can observe to the inhabit living in the soil. That is the decaying plants or animal material you can observe to the this type of the fungal you can observe the in this class. Another one, Zygomycotina. Important is the trichomycetes sexual reproduction to produce the spores also. Historic consideration medicine. Okay. The next one, coming to the another one, Ascomycetes. See here, this is a sac fungi that include molds and toughness of the baker's yeast. This is the Glamomycota also there. In the Ascomycotina, two best example is there. One is the yeast, second one is the penicillium. See, Glamiromycota, members of the Glamiromycota arbusculus. This is the mycorrhiza, one type of mycorrhiza. The roots are thalli. In the example is the bryophytes, land plants. Bryophyte is the land plants. You can observe the in the bryophytes, these plants are this type of fungi is present. Next one, this is the land plant. Geospina is the puriform forms cytobiotic association. In the roots, this fungi association with Nostoc, Anabina, cyanobacteria. Mostly, these are all the symbiotic systems. These are the all are called the symbiosis. Land plants for carbon and energy. And here main there is no recent circumstantial evidence are not there in this part. This is a gliospinopuriform. Walker and taken by the agricultural and food in Canada, you can use in this type. You can use the yeast also, no? Just like this part also. Another one, Pegigomycotina and penicillium. Very, very important. Tell me anyone who identify the penicillium? Which scientist? చెప్పండ్రా.
Alexander.
Penicillium ever identify చేశారు? Who's the first penicillium identify? The scientist name. Alexander Fleming.
Alexander Fleming.
Alexander Fleming, first in the separate from the fungal to this penicillium. Okay. In this one class is called Ascomycotina class. This is the Pejoja mycotina. It is the largest subphylum. This one is the largest subphylum of Ascomycotina. Includes the all filaments, sporocarps, medicinally very, very important in this one. Cocodes Valley, Fever Valley, saving drugs, penicillium, chrysogenus, this one. Last one, Saccharomycotina, Saccharomyces, yeast, baker's king under the yeast. This compresses a monophyletic lineage. With a single order, 1,000 known species are present. Here, 10 species account for the most plant mycotic infections, responsible for many biotechnological and industrial process to use for this Saccharomyces cerevisiae. Very important. This question to ask, industry and laboratory use for the fungi is the Saccharomyces cerevisiae is the laboratory uses for this fungi. Coming to the Basidiomycotina. Can I stop this one? Tomorrow I'll explain later. Yes? చెప్పండి. దివ్య, శైలజ, సంధ్య ఉన్నారు కదా, మాట్లాడండి.
ఎస్ మ్యామ్.
అర్థమైందా
ఓకే
ఈరోజు
మేడం.
క్లాసు?
ఎస్ మ్యామ్.
ఓకే, ఓన్లీ 1 మెంబరే రెస్పాండ్ అవుతున్నారు, మిగతా వాళ్ళు
ఓకే మేడం.
ఎక్కువ చెప్పినా మీకు బోర్ వస్తుంది, అవునా కదా? ఎస్ ఆర్ నో, టెల్ మీ.
మోస్ట్లీ ఎస్ మేడం.
ఏం లేదా? ఓకే, బోర్ ఏం లేదు, నాకే బోర్ వస్తుంది చెప్పడం. ఆ, అందరు స్కూల్లో ఉన్నారా, బయట ఉన్నారా?
అవుట్ సైడ్
మాట్లాడండి.
మేడం.
అవుట్ సైడ్ ఉన్నారా? మాయా, ఆర్ యు దేర్? మాయా, ప్రవీణ,
ఎస్ మ్యామ్.
ప్రవీణ భాయ్. ఓకే, డిడ్ యు అండర్స్టాండ్ టుడే క్లాస్? వాట్ ప్రవీణ?
మిడిల్ లో కనెక్ట్ అయింది మేడం.
మిడిల్ లో కనెక్ట్ అయ్యా, ఓకే. రేపు ఒక్క రోజు మష్రూమ్ గురించి చెప్పుకొని మళ్ళీ నేను అగైన్ నెక్స్ట్ సాటర్డే ఈ రిమైనింగ్ బెసిడియో మైకోటినా చెప్తా, ఓకేనా?
ఓకే మేడం.
సైమల్టేనియస్ గా స్టార్ట్ చేసుకుంటే కొంచెం అవుతుంది. రేపు అంతా మష్రూమ్స్ మీ కంటెంట్ లో ఉంది కదా, మష్రూమ్స్ గురించి కూడా.
ఎస్ మేడం. మష్రూమ్ కల్టివేషన్ యూనిట్ 5 మేడం.
అవునవును, ఎక్కడ ఉంది అంటే 1 సెకండ్. యూనిట్ 5 మష్రూమ్ కల్టివేషన్ ఉంది కదా, రేపు అది డిస్కస్ చేసుకుందాం. నెక్స్ట్ సాటర్డే ఈ క్లాసిఫికేషన్ వద్దాం, ఓకే?
ఓకే మేడం.
ఓకే, సీ యూ నెక్స్ట్ టుమారో సేమ్ టైం, ఓకే?
ఓకే మేడం. థ్యాంక్ యూ మేడం.
థ్యాంక్ యూ
ఎస్ మా, యా మా, ఓకే మా, బాయ్.
మ్యామ్.